Hello, my name is Joshua Mgavi. I welcome you to Reproduction in Living Organisms, questions 11 to 15. Question 11. Distinguish between oestrus and menstrual cycle. Outline the four main phases of the menstrual cycle. Describe the hormonal, physiological, and structure changes that occur during the human menstrual cycle. Part A. Distinguish between oestrus and menstrual cycle. Oestrus cycle, these are series of hormone-controlled changes in non-primate reproductive cycle, characterized by female experiencing a period of heightened sexual excitement just before ovulation. Menstrual cycle, these are series of hormonal controlled changes in primate female reproductive system. As a result, in monthly discharge of blood and uterine materials when fertilization fails. Outline the four main phases of the menstrual cycle. There is the follicular phase, ovulation phase, luteal phase, and menstruation. Part C. Describe the hormonal, physiological, and structure changes that occur during the human menstrual cycle. Gonadotrophin-releasing hormone from the hypothalamus stimulates the anterior pituitary gland to secrete the follicle-stimulating hormone. The follicle-stimulating hormone stimulates the development of the primary follicles in the ovary and also stimulates the walls of the ovarian follicles to secrete oestrogen. Oestrogen concentration in the blood rises quickly, inhibits the production and release of follicle-stimulating hormone by the anterior pituitary gland by a negative feedback mechanism. It causes the repair and the healing of the uterine wall following menstruation. Peaking of the oestrogen concentration triggers a certain surge in the production of both follicle-stimulating hormone and luteinizing hormone from the anterior pituitary gland. This triggers the ovulation and the remain of the Graafian follicle to develop into the corpus luteum, the yellow body. The corpus luteum stimulates the secretion of progesterone and estrogen. Progesterone, together with estrogen, they maintain the increase in thickness, muscularization, and vascularization of the uterus. They inhibit the release of luteinizing hormone and follicle-stimulating hormone by negative feedback mechanism. Decreased level of follicle-stimulating hormone prevent the development of the Graafian follicles. Hence, secretion of estrogen stops. Decreased level of luteinizing hormone prevent ovulation. Hence, the corpus luteum degenerates and progesterone decreases. Sudden decrease in progesterone level in the blood complete the menstrual cycle, as the hypothalamus secretes or resumes the secretion of gonadotrophin releasing hormone. Gonadotrophin releasing hormone stimulates the anterior pituitary to secrete follicle stimulating hormone as menstruation occurs, characterized by the breakdown and the shedding of the endometrial material. Question 12. Describe the structure of the anther of a flowering plant. Describe the events that leads to double fertilization in higher plants. 2. Ovulation. Part C. State the differences between stamens of insects and wind-pollinated flowers. Describe the structure of the anther of the flowering plant. The anthers, they are bilobed structures. They are bilobed structures. By bilobed, we mean that they have two structures on each side. Lobed, and then this side it is lobed, so we call them bilobed structures. Born at the slender flexible filament. Its transverse section reveals a butterfly-shaped structure having 4 pollen sacs bilaterally arranged in pairs with respect to the main axis. So when you look at it, it gives you a butterfly structure. And it has got 4 pollen sacs. It has 4 pollen sacs. There is this pollen sac here. That is the second one. There is a third one. And we have the 4th pollen sac also there. And these are bilaterally arranged in pairs. They are arranged in pairs with respect to the main axis. Pollen sacs contain several pollen grain from the pollen mother cells and is enclosed by the tapetum to provide nourishment to the pollen mother cell wall of the anther, are made up of fibrous layers with thickened cell walls, interrupted by lines of weaknesses called stromium, along which dehiscence occur. So you have the structures here. We have the pollen sacs. We have the pollen sacs. These are the pollen sacs which we have here. and our pollen sacs have got small, what we call mother cells. These ones inside are the ones that we call the mother cells. Now these mother cells are surrounded by a layer which we call tapetum. Surrounded by a layer which we call the tapetum, which provides nourishment to the mother cells of the anther, and it has got lines of weaknesses. It has got lines of weaknesses over which it breaks and then it discharges the seed content, or what we call the spore pollen grain content. The epidermis forms the outermost layer of the anther wall. Our epidermis, this outer layer, it is the one that forms the outermost anther walls. Along the midline axis lies the vesicular bundle made of the xylem and the phloem tissue. So in the middle, we have the xylem and the phloem. Those are the vesicular bundles in the middle, and continuous with the filament. So the vesicular bundle is continuous with the filament. Describe the events leading to double fertilization in higher plants. The pollen grains land on the stigma of a compatible species. The pollen grain will germinate, a process facilitated by sucrose solution, which is secreted by the epidermal cells of the stigma. Then the pollen tube begins to emerge. The tube nucleus occupies the position at the tip of the growing pollen tube. Meanwhile, the generative nucleus divides mitotically into a pair of male gamete nuclei. The pollen tube growth is positively chemotrophic and negatively urotrophic. Thus, grow down the style and grows towards the ovary. On reaching the ovary, the pollen tube enters the ovule through the chalaza, or what we call the micropyle, reaches the center of the ovule, penetrates the walls of the embryo sac, and bursts open. Tube nucleus then degenerates. One male nucleus fuses with the functional egg cell to form a diploid zygote. The other male nucleus fuses with the two polar nuclei to form a triploid primary endosperm nucleus. Part B. Describe the events leading to ovulation. The anterior lobe of the pituitary gland secretes the follicle-stimulating hormone, causing the growth of the Graafian follicle. It begins with the growth of the ogonium formed from the germinal epithelium into the primary oocyte, get enclosed into the Graafian follicle. The follicle-stimulating hormone also stimulates the release of oestrogen from the ovary, which causes healing and repair of the uterine wall following the previous menstruation. Increase in oestrogen level causes release of luteinizing hormone from the anterior pituitary gland. It surges, causes the rupture of the Graafian follicle to release the ovum into the oviduct. Part C. State the differences between the stamen of insects and wind-pollinated flowers. The stamens of insect-pollinated flower, the anthers are enclosed in the petal, whereas the stamen of wind-pollinated flowers, the anthers dangle outside the bract. Stamen of insect-pollinated flowers, they have short filaments. Whereas the stamen of wind-pollinated flowers, they have got long filaments and pedulars. The stamen of insect-pollinated flowers, they produce less pollen sacs in the anthers, whereas the stamen of wind-pollinated flower, they have got numerous pollen sacs in the anthers. Stamen of the insect-pollinated flower have fewer pollen grains produced, whereas stamens of wind-pollinated flowers, they have large quantities of pollen grains produced. The stamen of insect-pollinated flowers, the pollen grains are sticky, whereas the stamen of wind-pollinated flowers, the pollen grains are not sticky. Stamens of insect-pollinated flower, they are smooth and light pollen grains, whereas the stamens of wind-pollinated flowers, they have large and heavy pollen grains produced. The stamens of insect-pollinated flowers, they are brightly colored pollen grains, whereas the stamen of wind-pollinated flowers have dull colored pollen grains. Stamens of insect-pollinated flowers, the anthers are fixed at their base or fused along their backs to the filaments so that they are immovable. Stamens of wind-pollinated flowers, anthers are attached only at the midpoint to the tip of the filaments so that they can swing freely in air. Number 13. Distinguish between accessory sex organs and accessory sex character. Roman number 2, primary sex organ and secondary sex organ. Part B, mechanisms leading to fertilization and subsequent development of mammals are also evolutionally advantage to their success. Describe some of the mechanisms you consider are evolutionally advantage. Part C, outline the main features of sexual reproduction in mammal. Part A, number 1, distinguish between accessory sex organ and accessory sex character. Secondary sex organ or accessory organs are organs associated with the testes or ovaries, which play some role in reproduction, but other than gamete production and hormone secretion. For example, the penis, prostate, seminovesicles, sperm duct in males, and the fallopian tubes, uterus, vagina, mammary glands in female. Accessory or external sex characters are external characters which do not play a direct role in reproduction, but are distinct and enable sexes to be distinguished as male and female. For example, low-pitch voice and facial hair for males, high-pitch voice for females. Distinguish between primary sex organs and secondary sex organs. Primary sex organs produce gametes and secrete sex hormones, and development is under the control of follicle-stimulating hormone and luteinizing hormone. While the secondary sex organs do not produce gametes, they do not secrete sex hormones, and development is under the control of estrogen and progesterone in females and testosterone in males. Part B, mechanism leading to fertilization and subsequent development in mammals are of evolutionary advantage to their success. Describe some of the mechanisms you consider are evolutionary advantage. Fertilization and development are internal to limit wastage of gametes and provide protection to the young. The breeding seasons coincide with the breeding cycle, so birth occurs at a time when environmental conditions are most favorable for young growth. Feeding young ones or nutritious milk enables them to prepare for adult food as the digestive system develops. Secondary sexual characteristics enable easy identification of mating patterns. Parental care provides protection from predation and harsh environment conditions to the young. Development of the placenta enables gaseous exchange and the young to excrete wastes. Females are often more receptive to males during ovulation or the act of copulation stimulating ovulation. Fertilization is internal. Part C. Outline the main features of sexual reproduction in mammals. Females go through a sexual cycle known as the menstrual cycle. Sexual cycle is restricted to the breeding season, except in humans and other primates, which are sexually receptive throughout the year. Young ones are born at an advantaged stage. There is display of courtship behavior leading to mating. Development of embryo is internal and completely dependent on the mother for food and protection. The young feed on milk, and parental care to the young is prolonged. Question 14. Explain what is meant by pollinating organs. Explain the role played by pollinating organs in plants. Part C. Describe the artificial propagation methods in plants. D. Explain why root tubers are used as organ of vegetative propagation, yet their roots lack buds. Explain what is meant by pollinating organs. These are parts of the flowering plant specialized for vegetative propagation, as well as food storage, enabling plants bearing them to survive adverse environmental conditions. For example, cold or dry period. The food manufactured by photosynthesis in aerial green leaves is translocated and subsequently stored as starch in the rhizomes, culms, stems, and root tubers, or as glucose in onion bulbs. Explain the role played by the pollinating organs to the plant. During unfavorable conditions, the plant remains dormant, but when the condition normalizes, starch is hydrolyzed to sugar and translocated to the young buds, enabling early growth. Sprouting and photosynthesis, when they is little competition for nutrient and other species. Part C. Briefly describe the artificial propagation methods in plants. One is cutting, where a piece of root, for example, lemon, tamarind, or stems of sugarcane, cassava, or complete leaf is dipped in a rooting mixture composed of plant hormones and allowed to grow in a rooting composite soil. We have layering. This involves pegging down of the stem of runners, for example, strawberries, into the soil to induce development of the adventitious root, after which the new daughter plant are detached from the parent plant by cutting. We have grafting and budding. It is the insertion of a stem or bud in one plant, which we call the scion, onto another related plant, the stalk, ensuring that the vesicular tissues are connected. It propagates the lemons, the apples, roses, hibiscus, and the oranges. Part D. Explain why root tubers are used as organs of vegetative propagation, yet their roots lack buds. The root tubers and swollen taproots must bear a smaller part of the old stem if they are to act as organs of vegetative propagation. The swollen roots, together with the buds at the same base of the old stem, form organs of vegetative propagation and perennation. Question 15. Describe the mechanisms that promote outbreeding in monoecious plants. Part B. Explain how sexual reproduction may cause variation. Part C. With the aid of systematic diagram, explain how alternation of generation occurs in a sexually reproducing organism. Describe the mechanism that promote outbreeding in monoecious plants. They have separate males and female sex organs born on one flower or different flowers, but on the same hermaphrodite plant. Dichogamy. This is where the stamen and the carpels ripen at different times. Stamens ripening before the carpels, we call it protandry, or carpels ripening area is called as protagone. There is heterostyly. Here we have different style length. Structure of the flower when the stigma is protected from coming into contact with its own pollen. The stigma being taller than the surrounding stamen. Self-incompatibility. This is due to the genetically controlled production of chemicals that prevent pollen germination on the stigma of the same flower. Then there is self-sterility, in which the ovules cannot be fertilized by pollen from the same plant, thus calls for cross-pollination. In some flowers, the stamen hang out of the flower, and surely pollen is blown and scattered far away from the plant. Flowers possess brightly colored petals and sepals that produce nectar, have an attractive scent to attract insects, pollinators, which are agents of cross-pollination. Explain how sexual reproduction may cause variation. Crossing over. This is between chromatids of homologous chromosomes occurring during prophase 1. Linkage groups are formed at the chiasmata. New gene combinations created. We have independent assortment and random distribution of chromosomes. The orientation of the chromosomes on the equatorial plane during metaphase is purely random, allows independent assortment and separation during anaphase. Random fertilization. The fusion of the male and the female gamete is purely random, allowing varied characteristics in the offsprings. Then there is mutation, where we have chromosome and gene aberration. This creates new phenotypes in the population. We have artificial selection in form of polyploidy. Artificial fertilization have created variants of favored characteristics. Part C, with the aid of a systematic diagram, show how alternation of generation occur in a sexually producing organism. So when you look at this schematic diagram, you will see we have the sporophyte, which is diploid, 2N, and it is asexual reproduction. This one undergoes meiosis to produce spores, which are haploid. The spores germinate and form a gametophyte, which is N. Then the gametophyte will produce the male gametes, which are haploid, and also the female gametes, which are also haploid. And fertilization will take place. Then after fertilization, we shall form a zygote, which is a diploid, and that one will grow into a sporophyte, which is also diploid. Thank you very much for participating in reproduction in living organisms. Questions 11 to 15. Please do like, share, and subscribe.